Hi everybody, welcome back. Miss Calabrese here. In this video, we're going to be talking about the structure of cell membranes and transport across those cell membranes. All right, so first of all, just a general outline of what a cell looks like. So basic anatomy of a cell. All cells are going to have a plasma membrane. And then within that plasma membrane, we've got the cytoplasm, which is this like gel-like substance, which is cytosol, plus all of the organelles. And then the main organelle is the nucleus. So the nucleus is where we keep your genetic information. All right, so we're going to focus in on that plasma membrane and kind of understand the structure of it and how it functions. So the plasma membrane is made mostly of phospholipids, and it acts as this barrier between the inside of the cell and the outside of the cell. It's flexible, it's kind of floating, it's malleable, and it's semi-permeable, which means that it can choose which things come in and out of the cell. All right, so looking at the structure of it here, the main component are these phospholipids, and they form a bilayer, meaning two layers. They have a polar head. That's where the phosphate group is, and then the lipid tails are the hydrophobic part of the molecule. So those lipid tails, which are hydrophobic, kind of orient toward each other, while the phosphate polar heads create the outer and inner borders of the membrane itself. In addition to the phospholipids, we're also going to have some cholesterol molecules. So those cholesterol molecules are going to help add strength and stability to the membrane. So those are really important. And then we've also got some proteins. Right, so there are integral proteins, and those integral proteins span the entire lipid bilayer, so they go from one end to the other. And then we've also got peripheral proteins, and those are either attached to the outside membrane or to the inside membrane, but they don't go all the way through. All right, so main components there, phospholipid bilayer, cholesterol, and proteins. All right, so those proteins have a lot of different functions. So some of the proteins act as ion channels, which means they're little doorways that are going to allow ions to move in and out of the cell. Some of them are transporters, which also move things in and out of the cell in a different way. Receptors are proteins on the surface of the cell that are going to bind, usually with hormones, to change some sort of activity inside the cell. Enzymes we talked about previously, these are proteins that catalyze chemical reactions. And then we've also got some cell identity markers. These are glycoproteins, which means a sugar-protein combination that are on these are peripheral proteins on the outside of the cell that act as like identifiers or name tags. All right, so how do we get stuff inside and out of this phospholipid bilayer? And there are two major categories for transport in and out of the cell, and those are the passive processes, which don't require any energy, and the active processes that do require energy. So passive processes means that we're just moving across the cell membrane according to a concentration gradient, meaning substances tend to want to move from high concentration to low concentration. So they can just kind of slide down their concentration gradient without the input of energy. And active processes are going to require ATP. So sometimes that means moving from low to high concentration, and sometimes that means that we just have to change the shape of the membrane in order to get stuff in and out. All right, so as far as passive processes go, the main passive process is diffusion. We can also get passive transport through osmosis, which is the diffusion of water. Then we have facilitated diffusion. So facilitated diffusion is just like diffusion, except that we're using a channel protein to help facilitate that diffusion. And then filtration. And active processes, we're using these protein transporters that require energy. All right, so first to kind of understand how any of these processes work, we need to understand the concept of a solution. So a solution is a combination of a solvent and a solute. So the solvent is the thing that you're dissolving the solute in. So if I've got a sugar-water combination, water is the solvent, sugar is the solute, because water is the thing that's dissolving that solute. So in living organisms, water is the universal solvent. That's what we're kind of dissolving things into. And then things like salts and sugars and amino acids are going to be solutes. Okay, so starting with the process of diffusion here. So like I mentioned, this is just the process of particles wanting to go from high concentration to low concentration. So when we look at this picture here of the blue liquid, in that first picture A, you can see the blue particles in the liquid are all concentrated at the bottom of that graduated cylinder. So we've got a high concentration of blue particles down here and a low concentration of blue particles up here. So over time, we don't have to even add any energy to this. Over time, those blue particles are going to want to diffuse from high concentration to low concentration. So they basically want to spread themselves out and get as much space in between them as possible. And so at the end of that, that's going to yield an equilibrium solution where the particles are evenly spaced out. We've gone from the higher concentration to the lower concentration, and now we're all equally spread. All right, so that's the basic process of diffusion. Facilitated diffusion is a very similar process, except instead of the molecules just being able to pass across the membrane, they have to use a protein channel to help them get across. Right, so here's that concept of diffusion again. So if we see a high concentration of particles here on one side of the membrane, a low concentration of particles here on the other side of the membrane, these particles are going to tend to want to move from high concentration to low concentration. And this tends to just be really small particles. So small gases move this way. So this is how oxygen gets into your cells and carbon dioxide gets out of your cells. It just diffuses from high to low concentration by sort of squeezing right in between those phospholipids. All right, and then for facilitated diffusion, again, this is when we're using a protein to help us go again from high to low concentration. And there are two different ways that we can do this, either channel mediated or carrier mediated. Right, so here's a channel-mediated diffusion. It's basically just a gate, a doorway that you can open up. So this protein, this channel protein that sits in the membrane, acts as a doorway to allow these ions to move from one side to the other. And then carrier-mediated, similar, but it's a little bit more complex in that we need some sort of a lock and key in order to get this carrier to function. So we can transport glucose across a membrane with a carrier protein here, but we need a key in order to get that carrier to work. And in the case of glucose, the key is insulin, right? So we need insulin in order to get glucose to move across that membrane, even if we are going from high concentration to low concentration. All right, so there's those three concepts again. So simple diffusion, we're just kind of diffusing between the phospholipids from high concentration to low concentration. Channel mediated is when we've got this sort of open doorway, and that doorway will help whatever this particle is move from high to low concentration. And then the carrier mediated is when we need an assistant. We need an insulin molecule to help us go from high to low concentration. All right. Another type of passive transport here is dialysis. So dialysis is diffusion through an artificial selective barrier. For example, hemodialysis is the dialysis of blood, and it acts sort of like a filtration where we're selectively choosing which molecules are going to be able to pass through that barrier. And a lot of times dialysis is just selecting on the basis of size. So we'll move across the membrane from high concentration to low concentration if the particles are small enough to fit through the barrier. All right, and then osmosis. So this is another type of diffusion. This is a really specific type of diffusion. So this is the diffusion of water across a semi-permeable membrane. So I want you to really kind of drill that concept into your head, osmosis is the diffusion of water across a semi-permeable membrane, right? So it's when water is moving across the membrane to try to even out the concentration gradient. All right, so in order to kind of understand how osmosis works, we need to familiarize ourselves with the concept of tonicity. So this is going to refer to the concentration of salt, usually in solutions in the body. So that could be in the blood, could be in interstitial fluid, but we're talking about the concentration of a solute in the solvent, which is water. And then the cells are sitting in this water. The cells have semi-permeable membranes, and those cells are then going to try to even out the concentration gradient from the inside of the cell to the outside of the cell. All right, so an isotonic solution is one where we've got equal concentrations of salt on the inside of the cell as on the outside of the cell. So in your body, in your bloodstream, the isotonic amount of salt is 0.9%. So we got 0.9% salt on the inside of the cell, 0.9% salt on the outside of the cell. And that's going to mean that the water that's on the inside and on the outside of the cell is going to move equally in both directions. So we'll have water kind of moving into the cell, water moving outside of the cell, but osmosis is happening equally in both directions. So this is where the cell is happiest because the concentrations are even on either side of the membrane. In a hypotonic solution, the concentration of the solute is low in the outside of the cell and higher on the inside of the cell. All right, so this is a problem. So in this case, we can see that the salt on the inside of the cell, there's more salt on the inside than on the outside. We're going to assume that salt can't move easily across the membrane, but water can, right? Water can easily diffuse across that membrane, and that diffusion of water is called osmosis. So what's going to happen here is to try to even out the concentration gradient, since salt can't move and water can, water is going to chase after that salt to try to dilute it. So water is now going to rush into the cell at a higher rate than it's leaving the cell. Right, so that's what's happening in a hypotonic solution. So the cell is going to gain water and become big and round. This process, if we get in a solution that's too hypotonic, then we can result in hemolysis. So hemolysis means that that cell can actually burst because it takes on so much water. Right? And then the opposite situation of that is a hypertonic solution, so hyper meaning more. In this case, we've got a whole bunch of salt, really salty solution on the outside of the cell and not as much salt on the inside. So again, our concentration gradient is not even, but the salt can't move. So what's going to happen is water from the inside of the cell is going to rush out to try to dilute that outside solution. And that's going to result in crenation, which is the shriveling of the cell. Okay. Concept of filtration here. This is just the process. It's another passive process. This one works basically on the because of pressure. So if we've got hydrostatic pressure, water pressure that's pushing us through the membrane, that's essentially what filtration is. So, and this is important in your body because this is the main process that results in the formation of urine. Okay, so those are all our passive transport processes. So moving on to active transport. So this is your main active transport process that you're looking at in this illustration right here. So active transport is when we need to add energy in order to move things across the membrane. So what's happening here is we're moving particles against their concentration gradient. So we're taking stuff and instead of moving from high to low concentration, which is normal, passive easy, we're going to move from low to high. Right? So think of moving from low to high as climbing a mountain. You have to put energy in to push these particles up that mountain. Right? So if we're moving from low to high, we're going to need to use energy. And of course, our main form of energy in the cell is ATP. Right? So what you're looking at here is an ion pump, in particular the sodium-potassium pump. where if you pay ATP, you're going to be able to move ions across this pump. So what it does is it moves 3 sodium ions out of the cell for every 2 potassium ions it brings into the cell. So 3 sodiums go out, 2 potassiums go in, and we have to pay ATP in order to get that happen because there's more sodium on the outside of the cell and more potassium on the inside of the cell. So we're actually going against their concentration gradient. Right, so this is the main form of active transport. All right, another type of active transport, which really doesn't have anything to do with concentration gradients, is bulk transport, where we're using vesicles to move things in and out of the cell. So a vesicle is just kind of like a little phospholipid bubble that can carry stuff. So stuff can go travel around the cell on the inside of a vesicle. Um, Endocytosis is the process of bringing materials into the cell by vesicle formation. So we're changing the shape of the membrane, creating a vesicle and bringing something into the cell. And exocytosis is the process of getting something out of the cell by merging a vesicle with the membrane and then pushing the contents out. Right, so in endocytosis, we can subdivide into a couple categories. Phagocytosis, which is bringing in large solid particles, and then pinocytosis, which is usually bringing in liquids. All right, and so here's an illustration of what that phagocytosis looks like. So in this case, we're doing endocytosis. We're bringing, in this case, it looks like a bacterium into the cell. So this is something that your white blood cells would normally do. If they encounter a bacterium, they're going to try to do phagocytosis and bring it into the cell by the formation of a membrane. So we can see the membrane here is changing shape. We're taking that membrane and we're kind of wrapping it around the bacterium up here. And as we wrap the membrane around the bacterium, that's what's going to create this vesicle on the inside of the cell. We're going to call that vesicle a phagosome because it was the result of phagocytosis. Right, so that's how we're bringing something into the cell through vesicle formation. Pinocytosis is same concept, just a little bit smaller. So the vesicle we're forming is on a smaller scale and usually is only containing liquids. All right, so a couple questions just to review. What process usually involves movement of an item from an area of area of low concentration to an area of high concentration? So if we're going from low to high, what's that going to be? All right, and the answer there is if we're going from low to high and we need to add ATP, it's going to be active transport. All right, and next question here, what process is the movement from high concentration to low concentration with the help of a transport protein? So if we're using a protein to help us go from high to low concentration, what's that going to be? And of course, the answer there is facilitated diffusion. All right, so I hope this was helpful. Let me know if you have any questions.